Hi guys, how are you? I happy to be back and to talk to you today about chapter 4. So we're going to talk about the functional anatomy of prokaryotic cells. And you see that the eukaryotic cells I'm going to ask you to do as a homework. Since as we talked in the syllabus video, you will be expected to know some topics that you possibly learned in A&P, since this course is a continuation of our knowledge from A&P 1 and A&P 2. So, what is an eukaryotic cell? How it works? What are the structures? How protein synthesis work inside the cells? All this was A&P 1 content, so I expect you to know it, okay? So let's start as usually with our learning objectives. I'm gonna move my video here so we're not blocking anything. So for this chapter, you're gonna need to know the different bacterial shapes, structures of prokaryotes, including flagella, axiofilament, fimbriae, pili, what's the cell wall, how you stain cell wall, which are atypical cell walls, the lack of cell wall in mycoplasma, which is a species of bacterium, what's a plasmid nucleoid and endospore, and then we come to the eukaryote. So I posted 3 videos here for you to watch, which are related to protein synthesis in eukaryotes, DNA replication, and DNA structure. These are very important. I want you to be sure you understand this content, to understand how protein synthesis happens. What is the DNA? How DNA makes RNA? How RNA makes protein? What is the process? And if you have any question, come to my office hours and we can talk more about it, okay? So, the main differences between prokaryotes and eukaryotes. So what we really need to remember here is that, just getting my pen ready here, is that prokaryotes are going to have one circular chromosome that is not in a membrane. There are some exceptions of prokaryotes that don't have circular chromosome, but most of them do. So keep this in mind. The chromosome in the prokaryotes is circular. There's no histones. What are histones? These are the proteins that the DNA coils around to keep it organized in eukaryotes. So you don't have this in prokaryotes. No organelles. All those phospholipid bilayer enclosed vesicles, structures that you see in eukaryotes, you don't see in prokaryotes. Examples: chloroplasts, right, are organelles. So what else? Chloroplasts, mitochondria, all the vesicles like lysosomes, peroxisomes, endoplasmic reticulum, Golgi, all these organelles, the membrane, the nuclear envelope of the nucleus, none of that what's the name? I forgot. What is the name of that structure inside the nucleus? I just erased from my mind. But anything that has a membrane, very soon I'll remember. You will see, you will not see in prokaryotes, okay? Bacteria have peptidoglycan cell walls, okay? Archaea have pseudomurine cell walls. What else do we need to know? When you compare to eukaryotes, you have polysaccharide cell walls when they're present, usually in plants, and chitin cell wall in fungi, right? Division here is by binary fission. Most of the division in eukaryote is by mitosis, and we're going to add details later. So here I'm telling you what I want you to know from eukaryotes when you watch the videos, okay? I remember the word I was going to tell you that it's another organelle that is inside the nucleus. It's called nucleoli. I was thinking about nucleoid. Prokaryotes have a nucleoid, which is the region where the DNA is, but that's not an organelle. Nucleoli are present in, or nucleolus is the singular, are present in eukaryotes, and they contain the part of, it's where the ribosomal DNA and the ribosomal proteins are made. Ribosomes are present in prokaryotes because ribosomes are not organelles. So the ribosome is where protein synthesis happens, and they are present in eukaryotes. We're going to see that in a minute. So let's start talking about cell shape. So usually the average size of a bacterial cell is between 0.2 and 2 micrometers, okay, of diameter. And if they're round. If they're long, the diameter is around between 2 and 8 micrometers long. Some bacteria are pleomorphic. That means they can have different shapes in that same species. Okay, so they can be in one shape or a different shape. So you have, when it comes to shapes, we have bacillus, we have coccus or cocci, spirobacteria that can be Vibrio, Spirillum, or Spirochete. And I'm not going to give you species here because we're going to talk about the species later. Okay, so I'm just going to tell you the shapes. They can be star-shaped. These are very rare. They're not usually bacteria that cause disease that are star-shaped. And they can also be rectangular. So these are the shapes that we just talked about. Vibrio, Spirillum, Spirochete. You see how the Spirillum is much smaller than the Spirochete. Okay, actually, we don't have here exactly the same scale, but if you compare, so this is a bigger scale than this, right? So the Spirochete is actually smaller than the Spirillum. Cool. So this is a star-shaped bacteria, and this is a rectangular bacteria, okay? So how can they arrange? Bacteria can arrange as a duplet, or as a chain, or as a tetrad, or as what you call sarcini, is like their different axes when they divide, so they form this 8 structure cluster, or they can form these big clusters here that look like a grape cluster, and that's the staphylococci. So I want you to pay attention to this. When we talk about Staphylococcus as a species, you could have other bacteria that can have the same or resemble the same type of arrangement and are not Staphylococci. The same thing for Streptococci, okay? But most of the bacteria that form a strepto or a chain like this are from the Streptococcus genus. So those are for cocci. Bacilli can arrange together or not, so they can be single. They can look like a coccus but be a bacillus. This is a little difficult to identify, but it's a possibility. They can make a duplet, diplobacilli, or they can be streptobacilli. They can form a chain, and each cell, you see one cell here, each cell is rod-like, rod-shaped. All right, so now let's talk about the structure of a prokaryotic cell. So whatever you see here in red is going to be structures that are found in all bacteria. So all bacteria have a cytoplasm. The cytoplasm is all this that you see around the ribosomes. The ribosome are these little red dots, right? And then the plasma membrane is this yellow layer here. And then right after the plasma membrane, you have the cell wall, which is this dark brown layer here. The nucleoid is the region where you find the DNA. And the DNA is all this purple little spaghetti-like structure. And remember that the DNA is circular. So it's not going to have a blunt end. They're going to be bound together as a circle. You can see this here as a circle because it's kind of curvy, so you're not seeing the whole circle. But you have something, what else? Some bacteria have capsule, okay? Most of them do, but there are bacteria without capsule. So I'm not sure if capsule should be in red. Some bacteria may not have a capsule. We're gonna talk about what each one of these structure is for, okay? And then plasmids are found in some bacteria, and they usually contain genes that are related to production of or translation of a protein that is a toxin. Oops. Or genes for antimicrobial. This reads antimicrobial resistance. Okay, cool. So these type of genes are present in plasmids, and you see a plasmid here. What is this circular DNA? It's an extra chromosomal DNA, and this DNA is important because it can be transferred to other bacteria. So bacteria give pieces of their DNA to other bacteria, like a little gift. And the way these pieces of DNA are transferred, we're going to talk later, okay? Then you have the pilus talking about transfer. So pili, or pilus is the singular, is this structure that is responsible for transferring these little pieces of DNA from bacteria to bacteria, okay? Fimbriae. Fimbriae are these little hairy-like structures, and they are used, fimbriae here, and they are used for adhesion. And flagella, the long hair ones, that can be in different parts of the cell, are used for motility. But let's just look at them in just a little bit of detail. Glycocalyx. Glycocalyx is what makes the capsule. It's a layer of sugar. You're sugar coating your bacterium, right? So the capsule is the most external layer of the bacteria, and it's made of a sugar. It's viscous and it's gelatinous. See here? And it's made of polysaccharides and or polypeptides. What are polypeptides? Parts, pieces of proteins, right? So, and when the glycocalyx, so the glycocalyx is important for cell recognition. Each bacteria species has a different pattern of proteins and sugars in the glycocalyx. So it makes like their ID. They look different because their capsules are different, okay? The capsule is very organized and firm, attached around the cell wall. When that glycocalyx is unorganized and loose, you call it a slime layer. And slime layers are important for bacteria to form what is called a biofilm. We're going to talk about later too. So this second slide tells you that biofilms are made of slime layer, so they also are factors that contribute to virulence. Let's define virulence here. What's virulence? It's what makes a bacterium to be more pathogenic. So a virulent bacteria is a bacteria that is more prone to cause disease. Cool? So any type of glycocalyx, either capsules or slime layers, they are important to virulence. Capsules prevent the bacteria to be phagocytosed by phagocytes in the host body, and slime layers contribute to form those biofilms on surface. So it's a lot of bacteria together that is going to make them first more resistant to antibiotics because they talk to each other, they send chemical messengers to each other, and second because that slimy layer protects them, okay? This is capsule. See, the capsule here is this lighter part that you're seeing around the bacteria. Flagella are for motility, so they propel the bacteria, okay? So they can be organized in different ways. I'm going to give you an example here. So if they can be peritracheous when they're all over the cell. They're called monotracheous when you have only one flagellum in one of the poles of the cell. They can be called lophotracheous when you see just one big concentration of flagella on one pole of the cell. And when you see flagella on both sides of the cell, you call them amphitracheous. Okay? There are some slides in the book that show you how the flagella turns and what is the mechanism and the proteins involved. But all I want to tell you is that the basal body is embedded in the plasma membrane of the bacterium. Okay, so that's where the flagella is. There are flagella in eukaryotes. They have a slightly different structure molecularly, but remember, the sperm has flagellum. That's how the sperm moves. But it's a little different flagellum than the one that we see in bacteria. All right. What about axial filaments? Axial filament or endoflagella are, let me see if I get a different color here to show you better, are this little flagella that are along the body of spirochete bacteria. See? And this type of structure helps bacteria, for example, like the ones from the genus Leptospira, to move like a corkscrew. Okay, so that's the function of axial filaments. It's a flagellum that wraps around the body of a spirochete bacterium and makes them move like a corkscrew. Fimbriae. So this appendage, which is hair-like, allows for attachment. Is attachment important in pathogenesis? Answer 1, yes. Answer 2, no. What do you think? Totally. Think about how the bacteria is going to attach to the host if they don't have some type of adhesion strategy to be able not to fall from that host, right? So the more sticky they are, which is a feature that fimbriae give, the more pathogenic they are, okay? Because they're going to be able to stick to that host and not leave that host for a longer time. Pili, like I told you, are involved in motility, in gliding and twitching, but also in conjugation, where you have DNA being transferred from one cell to another, from one bacterium to the other. Okay, so the last thing we're going to talk here is the cell wall, and then I'm going to leave you and meet you in the second video, okay? So the cell wall of bacteria are made of peptidoglycan. Remember, we said that before, right? So when you see this picture here, the cell wall is this layer here before the capsule. You see how I'm lining the cell wall here? Cool. So what is the peptidoglycan? The peptidoglycan is a sugar mixed with pieces of protein. So what you have here is two different sugars called N-acetylglucosamine and N-acetylmuramic acid, NAG and NAM, that form what I like to call this brown and pink structure. I call it a candy cane. It looks like a piece of candy cane, right? And it's sugar. So all these bars of candy cane are connected together by peptides, by pieces of proteins, you see? So this makes a big mesh of pieces of protein, peptides, and sugar. And that's peptidoglycan. And that's what makes the cell wall of the bacteria. The difference between gram-positive and gram-negative bacteria is that gram-negative bacteria have a thin peptidoglycan layer, while the thick bacteria have the gram-positive have a thick peptidoglycan layer. Also, there are other molecules that are present in the gram-positive versus the gram-negative, and we're going to discuss that together with the staining of this positive and negative bacteria with the gram stain. in the next video. Okay, so I'll recap this in a minute with you. Bye bye.